welcome. In this video, we are going to talk about 3-point test cross of genetics. So, this is going to be a simple video because the topic doesn't consist of much. It's just about how to solve numericals and what is the main point of doing this 3-point test cross. What is the major reason, right? So, let's start to see what is a 3-point test cross. So, here we have a 3-point test cross is a cross involving 3 loci genes is referred as 3-point test cross. So it is simple that if a crossing over involves 3 loci, then it is referred as 3-point test cross, right? So it is used to determine the order and distance in map between each pair of genes. So what is saying that 3-point test cross helps us to determine the order of genes if they are in A, B, C in this manner, or it is A, C, B in this manner. So it tells us the order of the genes and the distance between A and C, C and B. It tells us the distance between those 3 genes, right? Either they are linked together, they are close by, or they are far away. In 3-point test cross, we get different recombinant products based on the crossing over, which should be single crossover or double crossover, right? So let's see. There is one chromosome with A, B, C in capital, and there is small a, small b, small c, 3 alleles, 3 recessive alleles on the other chromosome. So if no crossing over takes place, NCO, then parental type is formed. Now let's see about single crossover between A and B. If single crossover occurs between A and B genes, between the 2 chromosomes, so what we see is capital A, small b, small c. That is, it started from there, it took this one, and this took this one. So it formed this single crossover between B and C. So you can see A, B, and it came down small c, capital A, capital B, small c, while second one will be small a, small b, and capital C. So small a, small b, capital C. These are the 2 recombinant products. Then if we see double crossover between A and B gene and B and C gene, then what happens? It start with A, come down for B, and then go up for C again. So capital A, small b, and capital C. Here it will be small a goes up, capital B and small c, small a, capital B and small c. So about double crossover, what we can remember is only middle gene swap. In double crossover, DCO, middle gene changes, right? So it was capital A, capital B, capital C. So only the middle change, the capital B became small c, while this was small a, small b, small c, only the small b became capital B, right? So let's see the next one. Here we can see the criteria for 3-point cross, right? So what are the criteria for doing this crossing over? We already know that it is done to map, to know about the gene sequence and the distance between the genes, right? But what are the criteria for doing this cross? The first criteria is that the genotype of the organism must be heterozygous at all loci that will be used for the cross. Homologous alleles do not allow for differentiation of origin of alleles. Of course, they must be heterozygous at all loci. For example, let's see the earlier one again. So heterozygous, it is heterozygous. Of course, there is capital A, small b, so it is heterozygous. Again, this one capital B, small b, so of course it is heterozygous. Capital C, small c. So if it were homozygous, like if capital B, capital A, capital B, capital C, the lower one also had capital A, capital B, and capital C, then it would be a homozygous condition. But in such homozygous condition, 3-point test cross could not be done. 3-point test cross could not be done. That is because we need heterozygous to see if recombination had occurred or not, right? So for the recombinant product to be to be distinguished from the others, from the non-recombinant one, we have to have heterozygous condition. That is, both the two homologous chromosomes must have different alleles of the same gene, right? So let's see what is this is the first criteria. Then second criteria is the genotypes of the offspring must be able to observed in the order to determine which allele have been inherited, right? So for A, B and C and A, B and C heterozygous condition, if recombination had occurred and this double crossover occurred, and we find something like this: A, B, C and A, B, C. So in this condition, the middle gene have been swapped like it happens in double crossover. So in this condition, we have to have a different phenotypic difference as well. If the A1 is swapped, then single crossover, then a different kind of phenotype change must occur in the organism, so that we could identify that it is a parental type, it is a single crossover type, or it is a double crossover type. For us to understand if what it is genotypically, the offspring must be able to observed in the order to determine which allele has been inherited, right? So the genotype must be shown in its phenotype of the organism, so that we could identify which allele or where the crossover had occurred. Third criteria for 3-point test cross is that sufficient number of offspring must be produced in the mapping experiment to recover a representative sample for crossover classes. So this one is simple, that for us to understand that 3-point test crossover has occurred. So for us to find out all the recombinant products, like a few single recombinant products, double recombinant products, single recombinant between different genes. So for us to get all types of recombinants, we must have produced a sufficient number of offspring, a large number of offspring. The larger the number of offspring there will be, the better the idea of the data we will get, right? So that is important. Let's solve a question. There is this example AABBCC. Of course it's a organism genotype, and small a small bbcc. So this is the parental homozygous condition. Crossing over takes place. F1 is a heterozygous this genotype, F1 offspring. Then if we test cross, of course test cross means crossing with the recessive parent, right? So in test cross we cross the organism with its recessive parent. So in this case, this one is the recessive parent, so we cross this one with this one again, here this one. So this is test cross, right? So we observe something like this: parental type 305 and 310, as a single crossover between C and B, this many, and double crossover this many, single crossover between A and C this many, and total 1000, right? So what is the question? What is the order and linkage distance between gene A, B and C? Let's find that out. How will we solve this question? So what we are going to do is that we know that in double crossover, in DCO double crossover, what happens, that the middle gene is swapped, right? So we see this condition DCO. These are the two DCO. What is formed is A, B and small c, while in this condition it is A, B and capital C. That means C has been swapped, right? Here this is small c and there is capital C, so C has been swapped. So that means C is the middle gene. In DCO, we know that in double crossover only the middle gene is swapped. So the order of the gene will be ACB. ACB because the and ACB because the always in double crossover the middle gene is swapped, right? So this is the order of the gene. Now we have to find out the distance between the A, B and C genes. Let's see how will we do that. So order of the allele we have found out A, C, B. Now the distance influences the probability of crossover. We already know that if, for example, we take this example A, C, B and A, C, B, if this is, and if there is large number of crossing over between two genes, that means they are far away, right? But if there is no crossing over, very little crossover, then those genes are very close by. We already know this, right? So in this condition, the larger the distance, the more likely crossover will occur. The shorter the distance, crossover will occur less frequently. So determine the distance at the number of times of crossover occurred between the two loci. So this is the formula used to determine the distance between two genes. Number of single crossover between those two genes, number of double crossover between two genes are added, and then they are divided by the total number of offspring. So in this condition, and last at least into 100, right? So let's go back and see. So if we talk about the distance between A and C gene, between A and C gene is 42 and 43, right? So what we will have to do is that we have to add 42 plus 43 plus double crossover numbers. So double crossover numbers are of course 6 plus 9 plus 6 plus 9 divided by total number of offspring, that is 1000, right? And this total into 100. Let's see that again in the next slide. Distance between A and C, 42 plus 43 are the single crossover products of between A and C, and then 6 and 9 are the double crossover number divided by 1000 total number of offspring into 100. So this is equals to 10 centimorgan. The distance between gene A and C is 10 centimorgan. Now second is distance between C and B. So the single crossover number between C and B was 140 plus 145 plus 6 plus 9 are the double crossover numbers divided by total number of offspring, that is 1000 into 100, that is 30 centimorgan. The distance between C and B gene is 30 centimorgan. Of course, like I said earlier, the shorter the distance between the distance, crossing over will occur less frequently. You can see the distance between A and C is very low, so crossing over numbers is very low, while the distance between C and B is very high. That means they are far away. So here we see A and C 10 centimorgan away and C and B 30 centimorgan away, and the total side is 40 centimorgan. Let's see about another thing which is very important in such kind of question, that is interference and coincidence. So the crossing over is not uniform along the chromosome, right? Formation of one chiasma reduces the probability of another chiasma formation in immediately adjacent region of chromosome. This lack of independence is known as interference. So for example, we have these two chromosomes homologous A, B, C and A, B, C. So if there is a chiasma formation here during the crossing over, if there is a chiasma formation occurs here, so this chiasmata, this chiasma will reduce the probability of formation of another chiasma. That is, double crossing over is very less frequently seen, and the formation of one chiasma reduces the probability of another chiasma formation in the immediately adjacent region. This chiasma would not let any more crossing over occur here and here. In the adjacent region, one chiasma does not let the formation of other chiasmas. This lack of independence is known as interference. So this is interference, right? Now it was observed by H.J. Muller. This interference thing was observed by H.J. Muller. The net result of interference is observation of few double crossovers. The interference varies in different section of chromosome and is measured by coefficient of coincidence. So how would we quantify and measure this interference? So it is done by the coefficient of coincidence. That is, interference I is equals to 1 minus coefficient of coincidence. This is a formula you must remember. Now if the coefficient of coincidence is equal to 0, that is, coefficient becomes 0, so 1 minus 0 is equals to 1. That is complete complete interference. I becomes 1, right? If the coefficient of coincidence becomes 0, so 1 minus 0, that is, interference is equals to 1. So there is complete interference and no double crossover is formed. If the coefficient of coincidence between 0 and 1, it shows partial interference, and if coefficient of coincidence shows 1, that is 1 minus 1, then interference becomes 0. No interference, expected double crossover observed. Now there are some other formulas you need to remember in order to solve such question. There is coefficient of coincidence is equals to number of observed double crossover and divided by number of expected double crossover. Now we already know what are the number of observed double crossovers, but how will we find the number of expected double crossovers? Now expected double crossover frequency is equals to frequency in SCO region, I mean single crossover in first region, into frequency of single crossover in second region, and number of expected double crossover is equals to expected DCO frequency, this one, and into total number of progeny. So single crossover frequency between A and C we already saw 42, 43, single crossover, double crossover 6 plus 9. This was the SCO frequency between A and C, and divided by 1000, that is 0.1. While the SCO frequency, single crossover frequency between C and B was 140 plus 145, 6 and plus 9 divided by 1000, total number of progeny, gives us 0.3, which is the single crossover frequency between C and B. So we will put this in this formula. Expected DCO frequency is equal to frequency in SCO in first region, that is 0.1 into 0.3 frequency in SCO in second region. Thus expected DCO frequency is 0.03. Now we have to find the number of expected DCO. So that would be expected DCO frequency, which is 0.03 into total number of progeny into 1000, gives 30. Now number of observed DCO we've already seen, 6 plus 9 is equals to 15. Now coefficient of coincidence will be 15, that is number of observed DCO divided by 30 number of expected DCO gives 0.5. Coefficient of coincidence is 5, so I is equals to 1 minus 0.5, which gives us 0.5. I is 0.5, that is between 1 and 0 and 1, so there is partial interference. Some of double crossover will be seen. Now if fewer DCO than expected occur, I is a positive number, positive interference. If more DCO than expected occur, I is a negative interference. Thank you for watching. Please like and subscribe the channel. Thank you.